Hey everyone, welcome back. In the last segment, we finished up looking at signal transduction pathways that we label as STPs here. Again, three parts to it: reception, transduction, the change in shape of molecules inside the cell, and then some sort of cellular response. What we're going to do now is look at some different signal transduction pathways. All right, so all these pathways begin with reception. So a signaling molecule, often called a ligand, has a particular shape. And each ligand or signaling molecule docks on only certain types of receptors. So receptors, they are oftentimes in plasma membranes. That's why when we learn about the fluid mosaic model, all those different proteins embedded in cell membranes do different things, and some of them act as receptors. Okay, so some receptors are embedded in the membrane. Other receptors can actually be in the cytosol or in the cytoplasm. What will happen is, in many cases, the ligand will dock on the receptor, and then look at what happens. There's a shape change, right? And that shape change in the receptor is the first transduction, that first change that occurs. Now, some substances can actually diffuse through the membrane. So if you remember, the phospholipid bilayer has this nonpolar layer in the middle. So ligands that are small and nonpolar, so small, hydrophobic, or nonpolar, they can actually pass through the plasma membrane and dock on receptors inside the cells. If it's something that's large, charged, or polar, it won't be able to diffuse to the membrane, and it's going to have to dock on receptor proteins. So you can know a lot about the ligand based on where it's detected. If it's detected on the surface of the cell on a receptor protein, then you know it's large, charged, or polar. If the ligand is detected on a receptor in the cytosol, then you know it is going to be small and nonpolar, hydrophobic. So what we're going to do is look at three common types of membrane receptors. So these are proteins on plasma membrane surfaces that detect ligands and then cause signal transduction. So a very common one is a G protein coupled receptor. So here in green, that's the G protein coupled receptor. You have the nonpolar portions or regions of the protein embedded in the membrane here, and then you have polar portions. So a ligand, in this case some neurotransmitter, docks on the receptor, and then that communicates with a G protein inside the cell membrane. The G protein is either going to be in an active shape or an inactive shape, depending upon what gets bound to it, this GDP or GTP. I think it's like a guanine diphosphate or triphosphate. So here is the pathway. In step one, the G protein receptor is in an inactive shape. But if the ligand docks or the signaling molecule docks, look at what happens to the shape of the receptor. The shape of the receptor changes, so now it accepts the G protein. That's why they call it a G protein coupled receptor. So when the ligand docks, the receptor changes its shape. That's your first transduction. And then the G protein can dock there. When the G protein can dock, it can be activated by replacing GDP with GTP. When the G protein is now activated, it can head over to another enzyme embedded in the membrane. Oftentimes it's an enzyme called adenylyl cyclase. And look at what happens when the activated G protein docks on this membrane enzyme. The enzyme changes its shape from inactive to active, and then that further transduces the signal. It sends the signal on in the cell. Now, if you remove the ligand, then the receptor will go back to its original shape, and that can stop the signal. Okay? So again, it's all about interactions between molecules and shape changes. That's how communication occurs, and that's what allows your body to work, is the interactions of molecules resulting in shape changes. The next we're going to look at is receptor tyrosine kinases. And again, it's all about change and shape, right? So if you change shape, you can activate or deactivate different molecules. And an important component of this is phosphate groups. Think about the importance of ATP, right? Life doesn't exist without ATP providing energy. And all ATP does is hand phosphates off to molecules and change their shape and activate them. So as we look through these signal transduction pathways, pay attention to the phosphates, okay? When phosphate groups get handed to molecules, it changes their shape and activates them. So here are the receptor tyrosine kinases. You have a pair of them, and when the ligands dock, the receptors, it's called dimerize, they join together. So when the receptor proteins dimerize or join together, then it activates these tyrosines. Tyrosines type of an amino acid, so you can imagine how it will stick out on the proteins. And what's really cool about this is if there are multiple docking stations, once you phosphorylate this, you can actually stimulate multiple pathways. So one ligand, one type of signal, can actually lead to multiple responses within a cell. So you can activate different enzymes and get cells behaving in multiple ways. So this is sort of a fascinating way to sort of spread information through a cell. Another type of receptor in plasma membranes that we'll look at is a ligand-gated ion channel. This is going to be really important when we get into the nervous system, because what will happen is, again, a ligand docks on a receptor, and notice the shape change. This is called a ligand-gated ion channel. So there's a little gate that closes off the protein. When the ligand docks, it opens up the gate, and then the ions can diffuse down their electrochemical gradient. Two common ones are sodium and calcium ions. So the ions will flow down the gradient and then cause cellular responses. We'll get into how the nervous system uses the flow of sodium ions to actually cause action potentials and cause sort of rapid communication between cells. And then we'll also talk a little bit about calcium ions. All right, so those are all receptors that are on the plasma membrane surface. There are also certain types of receptors located in the cytoplasm. So if a signaling molecule is small and nonpolar, it can pass through the membranes and then dock on a receptor inside the cell. And then that activated receptor can actually potentially get inside the nucleus and activate genes. So nonpolar hormones that can get inside of nuclear membranes and activate genes can then start producing all sorts of proteins. And sometimes this can lead to long-term and permanent changes in the body. So think about like hormones of puberty. What will happen is certain hormones are produced, like estrogens and testosterone, and then those will actually get inside the nuclear membrane of cells and switch on genes, and you produce proteins that cause the secondary sex characteristics of males and females. So you can actually have signaling hormones activate genes inside of cells. Okay, we'll get into signal transduction here a little bit, and then we'll wrap up with one more video and some communication. Okay, looking at transduction, the actual shape changes that happen inside of molecules. One thing that is interesting about signal transduction pathways is that there's this notion of signal amplification. So if you activate a molecule or an enzyme, it has the potential to activate multiple other enzymes, which will then activate still more enzymes. And so every step in the pathway, you can activate multiple molecules, and that can amplify the signal. With signal amplification, and again, it's sort of the snowball effect. You can have just a small amount of signaling molecule lead to large-scale changes in a cell. And that's because as you have multiple steps, you can activate more and more molecules within each step. And it's actually helpful to have multiple steps in a pathway, because when we get into sort of coordinated response later on in the lecture, you can see how having multiple steps in the pathway provides multiple avenues for communicating sort of different responses or eliciting different responses in cells. All right, so phosphorylation is going to be key to this. We know when you add that phosphate group, you change the shape of molecules. So here, kinases will phosphorylate substances. So if you add a phosphate group, you will switch things on. If you remove the phosphate, you switch things off. So I think it's a simplified illustration, but I think it's helpful. So here you have some protein, and then if you phosphorylate it and add a phosphate group to it, it changes its shape and it can become active. Then if you dephosphorylate it, if you remove that phosphate group, it goes back to its inactive form. All right, and then you see that in these signal transduction pathways. So here's your ligand, there's your membrane receptor. It'll cause a shape change, activate proteins. And then what you'll do is have one enzyme activate subsequent enzymes in the signaling pathway, and on and on and on. Well, if you can phosphorylate, you have to also be able to dephosphorylate, and that's what these protein phosphatases do, the little PP protein. So what that'll do is actually remove the phosphate group from the kinase and switch the signal off. Okay, so it's important that if you switch a signal on, you also have the capacity to switch it off. Of course, you can imagine all the what ifs related to this, right? You think about all the steps in a signaling pathway and all the things that could go wrong, right? On the AP exam, who knows what signaling pathway they're going to give you? The important thing is to just follow the arrows and see what's going on and recognize that, okay, there's going to be a breakdown in the pathway. What if you have a mutation in the gene that produces these phosphatases that dephosphorylate these kinases? Well, if these phosphatases don't work, then the signal stays switched on and the response is continuous. Okay. If you have a breakdown in one of these kinases, so let's hear there's kinase 2. If you have a mutation in the gene for this kinase and you don't produce an active kinase, again, you can have the ligand, you can have the reception, you can begin transduction, but if you don't have the protein needed in the signaling pathway, that breaks down the signal. So I have no idea what they're going to show you on the AP exam. I'm just confident that you'll see some signaling pathway with arrows, and they're going to throw a wrench in it somewhere. Either something to dephosphorylate or phosphorylate something doesn't work, or maybe a kinase in the pathway is mutated and doesn't work, but something will happen to sort of throw things off. Okay. Ligands, we're familiar with ligands. Ligands dock on receptors. They're usually referred to as first messengers. They're the first signal. Second messengers are small, non-protein, water-soluble molecules or ions. Okay, so this is something inside the cell that basically sends the signal on. And the two most common ones that you'll see in signal transduction pathways, again, who knows what they'll give you on the exam, but two common ones are CAMP and calcium ions. CAMP is like cyclic adenosine monophosphate. So it's all just a derivation of molecules that exist in cells, like ATP, right? So if you convert the number of phosphate groups, you can make it AMP. So you have CAMP, the cyclic AMP, and calcium ions. So let's look at what goes on with that. CAMP is a second messenger that oftentimes activates an enzyme called adenylyl cyclase. We looked at that earlier here. So what will happen a lot of times is you'll have an enzyme like adenylyl cyclase embedded in the membrane, and what CAMP will do is actually activate it. So let's see what we're going with here. So this is a nice example. So here is your ligand. It's the first messenger, docks on the receptor. In this case, it's a G protein coupled receptor. That'll change shape, activate the G protein. G protein activates adenylyl cyclase, and then adenylyl cyclase converts ATP to the cyclic AMP, the cyclic adenosine monophosphate. And that acts as a second messenger to activate other kinases and elicit some sort of response. Well, what happens if you block adenylyl cyclase? If you block adenylyl cyclase, are you going to make CAMP? No. And if you don't make CAMP, what's going to happen to the whole pathway? It gets shut down, right? So again, you can think about if they give you some sort of like medication or drug that blocks some step in the pathway, it's basically going to shut down the signal. If you shut down the signal, you don't get the response. Calcium ions. Calcium ions are sometimes stored in the endoplasmic reticulum of cells. So, membranes act as containers, right? You have a plasma membrane surrounding the cell. It contains all the stuff inside of it. We've got these organelles inside of eukaryotic cells that also are bound with membranes. And then these organelles are basically sacs of different molecules. So we know in the mitochondria, we have enzymes related to cellular respiration, right? Well, in the endoplasmic reticulum, One thing that membrane-bound sac can store is a bunch of calcium ions. And then calcium ions can serve as second messengers in signaling pathways. So here, again, ligand docks on the receptor, activates the G protein. In this case, it activates another enzyme that cleaves this protein. That's a second messenger, which docks on a ligand-gated ion channel in the endoplasmic reticulum, and that releases calcium ions, and then the cell can respond in different ways. So again, do you need to memorize any specific molecule in a pathway? Nah. What you need to be able to do is just sort of follow the arrows and see the interactions. And again, when they give you a what if, predict what would happen. So what if there's a mutation in this ligand-gated ion channel here? Well, if it doesn't get the signal from IP3, then the calcium ions don't get released and the cell doesn't respond. Or what if someone takes a medication that mimics the shape of this IP3 molecule? Well, that would keep the ligand-gated channel open. And if you keep the ligand-gated channel open, then that causes the response to be continuous. There are certain neurotoxins. We'll get into it here shortly. Here, let's look at this. This is an example of muscle contraction. So when your muscles contract, there are two proteins that are doing that contraction. There's this thicker protein called myosin, and then a thinner protein here in orange called actin. Now you see the myosin protein, it has this, it's called a myosin head. What happens is with ATP, it rows. And when the myosin heads row, it pulls on actin, and then it pulls these proteins together. So that's what happens when your muscles contract. When your muscles contract, these myosin proteins are pulling on actin, and then it pulls the proteins together, and it shortens the proteins, the distance between these proteins, and that makes your muscles contract. Well, for the myosin heads to row, it has to have access to the orange actin proteins. And normally, these little gray ropes cover actin, so the myosin can't row. Well, what happens with your nervous system is when your brain sends a signal to your muscles to contract, there's this neurotransmitter, acetylcholine. It docks on receptors on your muscles. And then what will happen is you'll get an action potential, an electrical signal, travel through a specialized type of endoplasmic reticulum. This is in blue. This is called the sarcoplasmic reticulum. So again, this is a membranous sac that stores calcium ions. So when your brain is sending the signal to your muscles, that signal is acetylcholine. Acetylcholine causes an electrical change in your muscle cells that causes these ligand-gated ion channels to open in the sarcoplasmic reticulum. Calcium ions diffuse out, and then what those calcium ions do is pull back on those gray ropes. And when they pull back on the gray ropes, the muscles can contract. Okay, well, what would happen if a person takes a medication that does not allow these gates to close? If these gates stay open, then what happens is these ropes stay pulled back and the muscles will continue to contract. It's called tetany, or think of like tetanus, right? If you do not stop the signal, then with continued signals, the muscles can lock up or freeze. Or here, acetylcholine is the neurotransmitter. There's an enzyme that gets rid of this acetylcholine. It's called acetylcholinesterase. So there are certain drugs that are neurotoxins because what they do is inhibit acetylcholinesterase. So if you don't have acetylcholinesterase working, you basically keep acetylcholine docked on these receptors, and that will, again, keep the signal going. That keeps these gates open, and that keeps the muscles contracting. So if people are exposed to certain nerve agents or neurotoxins, you basically keep the signal going, the muscles lock up. That can impair breathing in people, and it can kill people. So all sorts of crazy things happen with signaling pathways. You have cells communicating with each other and starting and stopping signals all the time without us knowing about it. And without that, life doesn't happen. So signaling pathways are critical to our continued survival. Let's see. Well, I'll pause here and then we'll pick up in one last clip. Thanks for sticking with it.